In 2015, Dr. Tom Patterson and his wife, epidemiologist Steffanie Strathdee, were on holiday in Egypt when Tom became critically ill. His condition rapidly deteriorated, and he became insane with intense hallucinations. Steffanie contacted their trip insurance company, who arranged for a jet to fly them to Frankfurt, Germany, then another hospital a few days later. Patterson fell into a coma and was placed on a ventilator. At his worst, his body was pulling calcium from his bones and eating muscle to generate energy so he could survive, and he was on the verge of kidney failure. They did an endoscopy and discovered he had an abscess in his abdomen, a pseudo-cyst the size of a football, infected with Acinetobacter baumannii, a multidrug-resistant (MDR) infection that is notoriously difficult to treat. Initially, 3 antibiotics were given, but after 2 weeks, none of them had cured the Acinetobacter baumannii infection. He was transported back to the United States, and after repeated turns in Patterson's condition, Steffanie looked for alternative treatments and discovered bacteriophage. A bacteriophage is essentially a bacteria eater, named after the Greek word bacterion for bacteria and phagein, meaning to consume. They are viruses that infect and reproduce within bacteria. After discussions with clinical investigators, the FDA, and the Navy, the Pattersons were granted a compassionate use for bacteriophage therapy. Without FDA approval, the experimental therapy had to be used with special permission in the United States. She injected her husband with this virus, and his condition improved dramatically, and his recovery was almost miraculous. Bacteriophages are among the most widespread and diverse organisms in the biosphere. There are trillions of them, and they can be found anywhere bacteria exist, in sewers, rivers, nuts, fruits, food products, and even in your body. A typical phage has a head-tail structure. The head, shaped like a 20-sided cube, contains the phage's genetic material, while the protein tail attaches to the host bacterium to inject its DNA or RNA. The phage's enzymes break down the bacterial cell wall. Once inside, the phage's genetic material takes over the bacterial cell's machinery, redirecting it to produce viral components. The bacterial DNA is degraded, and the cell's resources are used to create new phage proteins and nucleic acids. New phage particles are assembled from the newly synthesized components. This includes forming the phage heads, tails, and tail fibers. The bacterial cell eventually bursts. For centuries, there has been a body of water in India that is highly revered and believed to have supernatural healing powers. In Hinduism, it's believed to have the power to cleanse souls. People worship and bathe themselves in this river. They drink its water for wellness, healing, and wholeness. And according to history, the results are instant. Now, in 1896, Ernest Hanbury Hankin discovered that there was something, he didn't quite know what it was, that could pass through a very fine porcelain filter and had a noticeable antibacterial action against cholera in Indian waters. Guess where he found it? The Ganges River. Decades later, in 1915, British bacteriologist Frederick Twort, who oversaw the London Brown Institution, found a tiny substance that both infected and killed bacteria. According to him, the agent had to be either a stage in the bacterial life cycle, an enzyme the bacteria made, or a virus that developed on the bacteria and killed them. He went on to name this virus bacteriophage, meaning bacteria eater. Unfortunately, the start of World War I, the lack of financing, and the discovery of antibiotics all interfered with Twort's research. Going forward, new research has revealed that bacteriophages may be one reason for the therapeutic nature of the Ganges River. Scientists have demonstrated that drug-resistant Pseudomonas aeruginosa infections can be defeated by bacteriophages found in the Ganges River's waters and is also beneficial for individuals with impaired immune systems. It has also been observed to be extremely selective, attacking particular bacterial strains while keeping beneficial bacteria intact. In 1919, a 12-year-old kid with severe dysentery was brought to a Parisian hospital. Félix Darel, who was working at the Pasteur Institute in Paris at the time, proposed phage therapy as a unique treatment for the youngster. This involved consuming a concoction of viruses that infect bacteria, that is, bacteriophages. At the time, medical professionals were hesitant to use this technique. There was very little proof of its effectiveness. There was no research on potential negative effects. Most significantly, it was still unknown whether bacteriophages actually existed. However, the boy's condition was getting worse. To prove the safety of the phage preparation, Durrell and a couple of his interns first drank it themselves during the strange events that occurred. The youngster received the preparation because there were no negative effects observed. Over the course of the following day, the boy's symptoms started to lessen and he quickly recovered. We're all thinking the same thing. Why are phages rarely used in modern medicine? The initial description of the virus in 1945 opened up new possibilities for treatment via phage. However, it was also around this period that the commercial manufacturing of penicillin began. This miracle medication was healing millions of people around the world. It offered so many advantages over the suggested phage therapy. Penicillin could be effective against a wide range of microorganisms. Phages, on the other hand, are known to be quite discriminating about the bacteria they infect. Penicillin was convenient to store and administer. The dosage can be easily standardized. Phage culture, storage, and dosage, on the other hand, were not standardized. There was little awareness about the potential negative effects of phage preparations. These variables instantly favored antibiotics over phages, bringing in the antibiotic golden age. Penicillin is an antibiotic that kills bacteria by stopping the formation of the bacterial cell wall, which is necessary for the cell's structural strength. It binds to transpeptidase, the enzyme that connects peptidoglycan chains in the bacterial cell wall. This binding reduces the enzyme's activity and prevents the production of a robust cell wall. The bacterial cell grows and expands, but the weakening cell wall cannot sustain the cell structure. The cell wall ruptures, allowing the cell to explode and die. When taken correctly, antibiotics can help kill bacteria that cause illness, but if you take an antibiotic while suffering from a viral infection such as the flu, the medication will have no effect on the viruses that are causing your illness. Instead, it cuts out a wide range of bacteria in your body, including some of the good bacteria that aid in digestion, infection resistance, and overall health. In fact, penicillin's abuse and misuse have led to the development of antibiotic-resistant bacteria, also called superbugs. Superbugs are germs that have become resistant to the drugs that should eradicate them. They're basically bacteria or fungi that have become immune to antibiotics. Bacteria in particular multiply very quickly. This allows them to take over the body's immune system and develop an infection. When a person takes an antibiotic, it fights and destroys the infectious microbes. However, they can adapt to their surroundings through DNA alterations. This alteration allows them to continue reproducing. While antibiotics kill the most susceptible germs, a small proportion of resistant organisms may survive the treatment. These stronger bacteria then multiply and take over, passing on their antibiotic resistance genes. These resistant bacteria can spread and produce infections that are more difficult to cure using typical antibiotics. Eventually, a person may develop an illness that is immune to the initial medication. The doctor may then try another antibiotic. Any bacteria that survives the second treatment may eventually develop resistance to it as well. As this cycle continues, the germ's resistance permits it to withstand a wide range of antibiotic treatments. Over time, it becomes a superbug. Each year, these drug-resistant bacteria infect more than 2 million people nationwide and kill at least 23,000 people in the United States. Due to this resistance, all antibiotics have been used, even carbapenems, which are referred to as the last resort medication. Some people fear that the present rate at which resistance is spreading could return modern medicine to the pre-antibiotic days. This means that steroid treatments, regular surgeries, or even a little wound could result in a fatal infection that is incurable. To stop this spread, we must use antibiotics as little as possible. The search for new antibiotics needs to be carried out. Unfortunately, in the last 30 years, just one new family of antibiotics has been identified. The World Health Organization repeatedly stated in a recent study the necessity for substitute agents capable of managing infections. Phage treatment is one of these interesting substitutes. For a couple of reasons, bacteriophage is rarely used in anti-infective treatments. When bacteriophages enter the human body, the immune system may recognize them as alien entities and initiate an immunity reaction. If they are used frequently or for a lengthy period of time, the human immune system may develop particular antibodies against them. These antibodies can bind to phages and use immune cells to destroy them or restrict their ability to infect bacteria. Likewise, phages are quite selective with the bacteria that they control. Targeting specific pathogens without compromising good bacteria is a benefit. However, treating illnesses resulting from several bacteria strains or situations where the precise bacterial root cause is unknown can be difficult with this approach. Despite these challenges, interest in phage therapy is growing, particularly as antibiotic-resistant bacteria become more prevalent. Improvements in fields such as genetic engineering, biotechnology, and a greater understanding of phage biology will eventually help to solve some of these barriers. As the years go by, phage therapy will keep presenting itself as a promising field for future medical treatments.